Okay, we're going to keep this fun train chugging along, and our next stop is the second half of the citric acid cycle. Our first learning goal is a continuation of last week's, illustrate the chemical logic and transformations that acetyl-CoA experiences throughout the citric acid cycle. Once we conclude the mechanisms of this wondrous cycle, we will be identifying its many modes of regulation. And at the very end, we're going to explain the intersection between the citric acid cycle and normal/abnormal cellular processes. We will be doing something a little bit different for the molecule of the moment. This is something that we'll be doing in class, and it'll be rather extensive. So we're going to put a pin in this for now. Picking up where we left off, alpha-ketoglutarate dehydrogenase complex catalyzes step 4 of the citric acid cycle. This is notable for two reasons. First, it is going to be our final oxidative decarboxylation step, and we can see that through the bird's eye view of the reaction. Alpha-ketoglutarate looks like so. This carboxyl group circled in red is going to be removed by this very complicated enzyme, and it will be replaced with coenzyme A, yielding succinyl-CoA plus 1 equivalent of CO2. While we do this decarboxylation, we will also be generating our second NADH equivalents, which we can see over here. So we're removing a carbon dioxide and getting a little bit of money in the bank at the same token. The third bout of chemical logic here is that notice how whenever we introduce acetyl-CoA, we're building in a thioester, shown over there. And that thioester product is going to greatly facilitate the mechanism of the next step. In regards to the mechanism, I've got some bad news and I've got some good news. The bad news is that its mechanism is practically identical to pyruvate dehydrogenase, which I know is a doozy. The good news is that you already are a master of the pyruvate dehydrogenase mechanism, so I don't have to torture you with it one more time. Moving past the mechanism, the last thing I want to mention is that this is the third regulatory step of the citric acid cycle because, again, it has a very negative delta G, negative 33.5 kilojoules per mole, which is made very negative in part thanks to this release of CO2, which is very entropically favored. And since this is such a negative delta G reaction, it's going to behoove us to insert a bunch of allosteric effectors, such as calcium, which will upregulate the activity of this step. And our allosteric deactivators consist of NADH and succinyl-CoA. Now, NADH and succinyl-CoA are products, which means this is, of course, a form of product inhibition. Step 5, succinyl-CoA synthetase performs the only instance of substrate-level phosphorylation within the citric acid cycle. Except here, we will be producing one equivalent of GTP instead of the ATP that you may be used to. And this enzyme achieves this by cleaving the thioester, high-energy thioester, within our coenzyme A moiety, liberating it, and in the process converting succinyl-CoA to just succinate. And the energetics of that cleavage happens to be roughly equivalent to the formation of one equivalent of GTP, which is why we make that little bit of energy right then and there. And in heavily catabolic tissues, such as like muscles, we do have isoforms of this enzyme that can take this GTP and then couple that to the formation of ATP by transferring that third gamma phosphate onto an ADP molecule. So the road can certainly lead to ATP synthesis. It's just not going to be in every cell. I'd like to re-emphasize that this is the only instance of substrate level phosphorylation that is going to occur in the citric acid cycle, or really throughout the aerobic respiration pathways in general. We will be using oxidative phosphorylation to make our ATP in the most part, but we can appreciate how we can abuse the high energy nature of this coenzyme A moiety by examining the mechanism, which we can boil down into 3 little steps. The first step of this mechanism is going to be binding of the substrate acetyl-CoA by the enzyme, and we are going to exchange that coenzyme A for a phosphate. So it's going to sneak in and cleave it off, giving us this enzyme-bound succinyl phosphate intermediate. The next step, there's going to be an active site histidine really close, catalysis by approximation, to that phosphate. The enzyme's histidine is going to then steal that phosphate right off of succinates, releasing it, but giving us this phosphohistidyl enzyme intermediate next. Well, now we need to get rid of that phosphate so we can start this cycle over again. And it would be such a shame if we just hydrolyzed it with water. What if we employed GDP to pick it up instead? And that will, of course, leave us with GTP. So GDP steals that phosphoryl histidine, or the phosphate off of that histidine, and then we can go on our merry way, repeating the enzyme cycle. Now that we have liberated succinates, which is a 4-carbon acid, from succinyl-CoA, the goal of the citric acid cycle moving forward in this last third is going to be getting back to oxaloacetates, OAA, which is also a 4-carbon compound. Now, why are we trying to get to oxaloacetate? Well, if you recall, the citric acid cycle begins with the combination of oxaloacetate and incoming acetyl-CoA to form citric acid. So we need to get to oxaloacetate if we want this reaction cycle to continue on and on and on again. The enzyme that's going to catalyze step 6, succinate dehydrogenase, is also a really interesting one for a couple of reasons. Let's talk about succinate dehydrogenase's chemical logic first. It's twofold. Step 1, or part 1, it's going to be the sole instance of oxidation by FAD. All right. The cofactor you're probably used to at this point is NAD+. That is the entity that is removing hydrides from our metabolites. Now we're going to change things up and use FAD to form one equivalent of FADH2. And there actually is a good reason for that. You're going to see this when we talk about oxidative phosphorylation, which is down the road. But all you need to know for now, though, is that FADH2, like NADH, is money in the bank. We're going to cash that in for a lot of ATP really soon. As a byproduct of making this FADH2, we will also be obtaining one equivalent of fumarates, all right? And we again form this by removing these hydrides through a redox process, dehydrogenase. Once we dehydrogenate those red hydrides, we will be left with an alkene between carbons 2 and 3 within the fumarate molecule. So why are we making the alkene? Well, that's also going to be useful right here, right now, because in the next two steps, we are going to be working towards oxidizing it one last time to get a little bit more money into our bank. And alkenes are going to be more reactive than alkanes. Now, what's another cool thing about succinate dehydrogenase? This is another great example of an enzyme that does moonlighting. All right, protein that has two pretty different roles in its repertoire. All right. Succinate dehydrogenase is interesting because it is a membrane-bound enzyme. The mitochondrion, it has an outer membrane. It has an inner membrane. And the inside, the matrix, is over here about. Our step 6 enzyme is actually anchored within the inner membrane. This little part protruding from that inner membrane is the one that's going to take in succinate and convert it into fumarate. The part of the protein that is embedded within that inner membrane, well, that is actually going to be a whole different kettle of fish, if you will. That part of the enzyme is actually participating in the electron transport chain, which is needed for oxidative phosphorylation. We're going to talk about that and its mechanism later on. But I would just like you to know that the FADH that we create through this oxidation, it's not just released into the matrix. It gets used right away in this membrane-bound portion. Which I've circled over there. Okay, so step 7 is the second to last step of the citric acid cycle. It's catalyzed by the enzyme fumarase. And what fumarase does is take our alkene diacid, fumarate, and it will hydrate it with an activated water molecule. And when we do that, when we add an OH to a double bond, what's the result of that? We're going to get an alcohol, specifically L-malates. What is the chemical logic of this mechanism? Well, what we're doing is hydrating an alkene to form an alcohol. And alcohols can be oxidized pretty easily by things like, I don't know, NAD+, right? Something else you should know about fumarase is that it is a very efficient enzyme. It is also one of those catalytically perfect entities, and it's actually so good at its job, we haven't done a terrific job of understanding its mechanism because that's how fast it goes. All I really want you to know is that fumarase is going to be a highly stereospecific enzyme. It will not hydrate cis fumarate. That will actually competitively inhibit it. It only works on trans fumarate. See how these carboxyls are on the opposite side of that double bond? It's trans. Okay, we made it, the final step of the citric acid cycle. Step 8 is catalyzed by the enzyme malate dehydrogenase, and the fact that it's a dehydrogenase should cue you in by now that we are removing a hydride to reduce a substrate, specifically NAD+, to form NADH, money in the bank. That is going to be the first instance of chemical logic of this transformation. And look how convenient this is. When we do this redox reaction, we not only get energy out of L-malate one more time, but we end up with oxaloacetates. This regeneration of OAA is what makes a citric acid cycle a cycle. We begin right where we ended off from, and it's going to be a nice, neat little circle. You're probably looking at the delta G of this reaction and thinking, holy smokes, positive 29.7 kilojoules per mole. That's a rather uphill transformation. So you may think that this final step is going to be blatantly favoring the reverse reaction or the reformation of malate. However, we have thermodynamics on our side here. Remember step 1, which is an incredibly irreversible regulatory step, citrate synthase. The irreversibility of that first transformation prevents the accumulation of oxaloacetates. So this is not going to linger within the mitochondrial matrix, at least under catabolic conditions. And that is what's going to help drive this reaction forward, even though it is quite energetically uphill. It is actually the most energetically uphill reaction of the entire citric acid cycle. As for the mechanism, I've already showed you actually how NAD+ oxidizes alcohols, so pat yourself on the back. You have just survived another 8 mechanisms. Let's take a step back and summarize what we've learned. The citric acid cycle, its job is to fully extract the chemical energy out of acetyl-CoA. And what do we get when we do this? 3 equivalents of NADH, 1 equivalent of FADH2, and then 1 GTP or ATP. It depends on if this is happening like in a muscle cell or not. So this is the energy that we've extracted here. And at first, this may feel a little bit underwhelming. I mean, 3 NADH, 1 FADH2. And what are we going to do with that? Well, that's coming up when we talk about oxidative phosphorylation. Remember, this is money in the bank. And we are going to cash this in with plenty of interest and get a whole bunch of ATP once we do the third and final step to respiration. Also keep in mind, all of this is occurring once per acetyl-CoA. We ultimately derive 2 acetyl-CoA molecules from 1 molecule of glucose, so all of these benefits get doubled per sugar molecule. Another key takeaway here is that 100% the citric acid cycle is considered firmly in the realm of aerobic respiration. However, did we use oxygen once in this entire conglomerate of enzyme craziness? We haven't, but that is actually going to come later. We're going to use oxygen towards the very end of the last phase of aerobic respiration. However, we have to realize, though, that we do not want to make all of these NADHs and FADH2s if we do not have the oxygen available to cash in our ATP prize. So there needs to be communication between the citric acid cycle and really the rest of the cell so that we can work together and make our money in the bank molecules wisely. And the way we achieve that is through common effectors. What do I mean by that? Well, both glycolysis and the citric acid cycle, for example, are particularly sensitive to NADH and ATP. Study these graphics really well and look at all the enzymes here that are sensitive to energy charge. We have ATP being an inhibitor for the PDH complex. It's going to inhibit citrate synthase, our first regulatory enzyme, oh look, it's going to inhibit our second regulatory enzyme, and that looks like that's all the instances of ATP inhibition. All right, now let's take a look at NADH. This is produced by glycolysis and the citric acid cycle. If it accumulates, where is it going to shut down? PDH, citrate synthase, and alpha-ketoglutarate dehydrogenase complex. So we not only get punished by accumulating these unspent energy molecules, but we get punished in many different areas, which is going to bring the traffic of this whole process to a halt. The citric acid cycle also has relevance to cancer. If you remember how cancer has that sweet tooth, well, loss of function mutations in fumarate and succinate dehydrogenase, these are going to promote oncogenesis, and it will do so through two different reasons. Number one, that's going to force the cell to uptick glycolysis. If aerobic respiration machinery is broken, we need to abuse the glycolytic pathway to compensate, and cancer loves glycolysis. The second perk for cancer, I suppose, is that breaking fumarase and succinate dehydrogenase will cause an accumulation of fumarate and succinate. What will happen is those two substrates will accumulate within the mitochondrion and they will leak out. And they just so happen to inhibit the proteins that inhibit hypoxia-inducible factor. So these two inhibit HIF inhibitors. So that can be kind of confusing to keep in your head, but I'll remind you that hypoxia-inducible factor promotes glycolysis. If we're inhibiting the inhibitor to glycolysis, we are going to be accelerating it. Now, likewise, certain cancers actually employ a gain of function mutation to specifically the enzyme isocitrate dehydrogenase. And this is really insidious. What it's going to do is change its functionality so that it converts alpha-ketoglutarate into not isocitrate, but this other substrate called 2-hydroxyglutarate. This is a problem because 2-hydroxyglutarate will inhibit certain histone demethylases that are responsible for silencing the genes that regulate the cell cycle. Cell cycle regulation genes silenced. So the cell is now at liberty to divide without any kind of inhibition. Very briefly, I'll mention that the citric acid cycle is amphibolic too. It is catabolic, meaning that it produces energy, namely these NADH and FADH2 molecules. However, we're also going to make these many pretty metabolites that the cell can use for anabolism. Citrates, we will learn soon enough, will bleed out into the cell and stimulate fatty acid synthesis and lipid synthesis. Oxaloacetate, we've already seen that that can be bled out and converted into glucose via gluconeogenesis. And look at this. You can have alpha-ketoglutarate become a number of amino acids and nucleotides. Acetyl-CoA is how we get porphyrins and heme. So these metabolites, they aren't just earmarked for energy production. They're super useful for a number of other metabolisms. The last item I'll highlight is that as complex as this material is, our knowledge of the citric acid cycle is still growing. Doesn't it seem a little bit, I don't know, hard to believe that citrate just bounces blindly and happily across 8 different enzymes in a perfect loop without any mistakes every time? Well, some very smart enzyme kineticists were able to use math to determine that it's actually impossible to have the substrates randomly diffuse fast enough across all 8 enzymes in a way that would produce enough energy to power life. So for decades, scientists hypothesized that the citric acid cycle enzymes actually need to work together, kind of like pyruvate dehydrogenase complex. Scientists proposed that these citric acid cycle components would form transient, complex quaternary structures in times of high demand, so it would help channel the citric acid cycle intermediates from one reaction to the next, to the next, to the next. And the word for these temporary constructs is metabolons. And I want you to think of metabolons as assembly lines. They're going to line up the enzymes head to tail and just pass the molecules through, which is going to streamline all of these complicated transformations. And the fact that they form reversibly and through weak interactions makes them really difficult to study because it's going to be hard to get a picture of them if they're constantly forming and breaking. Now, I learned about metabolons when I took biochemistry back in 2014, and they were still theoretical back then. But the cool thing about science is that, well, mere months after I took biochemistry, we published the first structure of a metabolon that was discovered using electron microscopy. Electron microscopy, if you don't know, is a very powerful microscopic technique, and we finally got it good enough that was able to resolve very large molecules, like complexes of proteins. And remember that metabolons come together and break apart. So what these scientists did in this paper, which is one of several on the first metabolon discovered in the citric acid cycle, they added a cross-linking reagent, disacinamidyl glutarate, which glued the pieces of the assembly line together. These two succinimide groups are really great leaving groups. So nucleophilic lysines of the metabolic machinery would displace these two portions, and that's going to cross-link and glue the proteins together of the metabolon. So what does that metabolon look like? Well, here is the molecular model over here. This is a complex structure of malate dehydrogenase, citrate synthase, and aconitase. And those correspond to enzyme number 8, 1, and 2 of the citric acid cycle. That right there is 1/3 of the citric acid cycle all lined up. And it gets even spookier than that. The active sites of these 3 enzymes are linked together by a valley of positively charged amino acids. This creates a highway for the negatively charged intermediates to drive along, which reduces reaction entropy. And the crazy thing is, we're actually discovering more and more metabolons all throughout biochemistry. There's glycolytic metabolons that have been discovered. There's metabolons for lipid metabolism too, and there's probably several others that I haven't read of yet. And to think that the existence of metabolons was, frankly, a well-educated guess for almost 45 years, and now we're seeing that they're all over biochemistry is just so impressive. And as a bonus, I'll throw in this nice summary slide that gives you the spark notes for the 8 reactions that constitutes the citric acid cycle, a one-sentence summary of the chemistry that each entails, along with the key metabolites that get generated with each reaction. So good luck, and I hope this helps you study.